Hi students, I want to give you a brief introduction to this idea of Hardy-Weinberg. So Hardy-Weinberg, these are two scientists that got together and they've created this useful tool. It's a mathematical model that allows us to look at allelic frequencies, so the frequency of the dominant allele and the recessive allele in a population, and make predictions about what future generations should look like. And so I'm going to go through each of the components of this mathematical formula step by step. So let's say we have a population of birds, and some of the birds are black and some of the birds are blue. And let's say that the black coloration is dominant to the blue coloration. So looking at these birds, we might wonder what their genotypes are. So if we looked inside at their genotypes, we might see something like this. So let's say that like I said, black is dominant. So if you're looking at the blackbirds, you'll see that some of them are homozygous dominant, big A, and some of them are heterozygous, big A, little a. And both of those would be black. And you'll notice that all of the bluebirds are homozygous recessive, because that's the only way you can get the blue coloration. So that's what their alleles look like in their genotypes. We pulled all the alleles out so we can see them. So here they are. Here are all the alleles. If this is a randomly reproducing population, that they randomly mate with one another, then all their egg and sperm are going to be dumped, quote unquote, into a bucket, which we call the gene pool. And so if we're looking in this gene pool, how many of the actual alleles, the actual genes, are dominant genes, and how many of the actual genes are recessive genes? So looking at the big A's, here's all the big A's. So obviously the homozygous dominant ones have 2 of them, but the heterozygotes are also carrying 1 big A. And if we counted them up, we'd find that 11 out of the 40 letters represented there, or alleles, are big A. And so that gives us 0.275 or 27.5%. And then necessarily the little a's are the other 29 out of the 40 letters, or 72.5%. So in this gene pool, 27.5% of the alleles are dominant and 72.5% are recessive. So if we were randomly make egg and sperm out of this and mate them together, this is what's generally happening in a randomly mating population. And you'll notice that of these egg and sperm, that about 27% are big A's and 72% are little a's. And so what are the chances of getting an offspring that gets 2 big A's, or is homozygous dominant? Well, if it's just simple probability and we reach our hand into the gene pool and grab a random egg and grab a random sperm, then the chances of getting 2 big A's would be 0.275 times 0.275, or 7.6%. And likewise, what are the chances of getting a little a offspring, or homozygous recessive? Same thing. The chances of grabbing a recessive are 72.5%, so 0.725 times 0.725 ends up being about around 52.6% that would be homozygous recessive. Now, let's look at what are the chances of getting an offspring that is heterozygous, gets 1 big A and 1 little a. Well, the chances of pulling a big A are 0.275 and then a little a, 0.725. So that gives us about a 19.9% chance of getting big A then little a. But you'll notice you could also grab a little a then a big A, which is also 19.9%. And since either one will be a heterozygote, we're going to add those together. And so we have 39.8% that will be heterozygous, given the allele frequencies in the population. So let's write a generic formula for everything I just said. So again, if we look at the egg and sperm and we notice that 27.5% of the gametes are dominant and 72.5% are carrying that recessive allele, we're going to generically label the dominant frequency P for probability. And we're going to randomly label the recessive frequency Q. And that's just a mathematical convention when you use P. Q is the other letter that we often use. And so those are our dominant and our recessive. And so then what are the chances, looking at the population, that a blackbird is homozygous dominant? Well, remember what we did. We took the probability of the big A and we multiplied it together, so P times P. So P squared would represent the probability of randomly reaching into the gene pool and getting 2 big A's in our offspring. Same thing for big A, little a, you would do P times Q or Q times P. Either one works, so it's 2 PQs. And then for a homozygous recessive, it's Q times Q, or Q squared. And so this allows us, if we know the frequency of the alleles in the population, we can predict the genotypes of the offspring, and therefore the phenotypes of the offspring. Or vice versa, if we know the genotype or phenotype frequencies in the offspring, then we can make an estimate of what the allele frequencies are in the gene pool. And then if something happens, if the population is evolving in some way, these allele frequencies will change, and then we can make some predictions about what's changing. And so that's the definition of evolution, right? Change in allele frequencies. And this is how we calculate allele frequencies.